Question 1 asks you to describe how the stages of mitosis result in the formation of two diploid cells. This is quite a loaded question, but if you focus on the important words, it's actually very straightforward to answer. Effectively, it's saying describe the stages of mitosis. There's nothing unique about the formation of two diploid cells. Mitosis always produces two diploid cells from one parent cell. A diploid cell is a body cell as opposed to a haploid cell, which is a sex cell. This question is actually one of the easiest 6-mark questions you can answer, providing you've memorized the information. There's no application, which keeps it quite simple. So firstly, you need to know the stages of mitosis, of which there are 4. There is prophase, metaphase, anaphase, telophase, and cytokinesis, which is essentially part of telophase. If you remember PMAT, that should help. You were also given pictures here to give you a prompt, although they're not fully detailed, and your answer requires you to go into a little bit more detail than is shown in these pictures. So these pictures show you prophase over here, and you can think of P for prepare, because the cell prepares to divide. Then metaphase, where the chromosomes meet in the middle of the cell, M for middle. Then anaphase, where they're pulled apart to either side of the cell, apart for anaphase. And telophase, which is the terminal phase, the final phase, where the cell actually divides. So all you have to do now is describe 2 things roughly that happen in each phase. So in prophase, the nuclear membrane disappears and the DNA unwinds and is condensed into chromosomes. So the diagram doesn't help you too much here. You can see that the DNA here is becoming more distinct as it shortens and fattens. This is what we mean by condenses. So the DNA condenses, and for the first time you can start to see them as chromosome-like structures. And they don't show you the nuclear membrane, so the nucleus has a membrane and that disappears. They don't show you that, so you need to remember that. In metaphase, chromosomes line up along the equator of the cell and spindle fibers attach to the centromeres. So the diagram doesn't show you this fully either. It shows you chromosomes lining up in the middle of the cell, but you can't see the spindle fibers, so I've drawn them in, and they will attach to chromosomes. So before division, this is what a chromosome looks like, and as it prepares, by the end of prophase, it looks like this. Basically, it's 2 of exactly the same chromosome attached together at a hinge point. This hinge point is called the centromere. In this form, we refer to each individual chromosome, which is a replicate of the other, as a chromatid, though they're genetically identical. So the spindle fibers basically attach to the centromeres. And this region, which we call the equator of the cell, like the equator of the Earth, the center of the cell, that's where they'll meet in the middle. And in mitosis, they sort of line up single file, but you can't see that here very clearly. So just relate back to the point, chromosomes line up along the equator of the cell here, and spindle fibers attach to the centromeres. You can't see that, but I've drawn that in here, and they're attaching to these parts of the chromosomes. During anaphase, the apart phase, the spindle fibers contract. That means they start to shorten, they start to pull in, pulling the chromatids to either side of the cell. So you can see them moving to either pole of the cell. So spindle fibers contract, pulling the chromatids to the edges of the cell. A little tip for you, in an exam, if they ever ask you to recall the stages of mitosis from a picture, they normally either focus on metaphase or anaphase, so it's well worth remembering what the images look like. Finally, in telophase, the nuclear envelope forms around each set of chromosomes. The picture doesn't show you that, so I've drawn them in here. And that separates them from one another. And finally, cytokinesis occurs, dividing the cytoplasm, creating 2 diploid cells. Cytokinesis is basically the division of the cytoplasm, the jelly-like fluid that is inside a cell. So that divides, a membrane sort of seals it off, and you get 2 diploid cells. So remember, this is just describing the stages of mitosis. There's nothing to apply here. You just remember what happens in each stage. If you don't remember it, you can't answer it. But for that reason, it's quite an easy question as well, because it's just testing your memory.